So just checking that are we ready to go again?
Yes.
Okay, right. So part 2 then. So in the second part, I'm telling you about some of the work that we've been doing under this GplusE project. So again, divided it into 3 sections, and the topics initially just generally looking at using transcriptomics to investigate factors affecting fertility. And then the data have been analysed in various different ways, and I'm just going to tell you about the results of 2 analyses. One was looking at correlations with IGF-1, and the other was looking at differential expression according to fertility. So just to pick up where we left off from the previous talk, so a sequence of events around calving. So one of the points I made to you when we were talking about some of the things that affect what happens during lactation and that affects fertility is that a lot of it actually starts pre-calving. So during that dry period, the cow may have had mastitis in the previous lactation, there may be pre-existing inflammation, and depending on the nutritional status, lipid mobilization may well begin before calving, and as we saw with some of those earlier slides, it may also enter the calving period with reduced immunity. And all of these things are going to increase the likelihood of calving problems. So together with those and what's gone on before, that all affects the increased likelihood of a metabolic imbalance, increasing the likelihood of clinical diseases and reducing the likelihood of conception. Just looking at the graph, again looking at this negative energy balance situation. So once it has calved, milk production rises to a peak, but the dry matter intake lags somewhat behind it. So during this early lactation period, the cow is using body condition for milk production, and that leads to this decrease in body weight. So in terms of the GplusE project, Michelle's already essentially shown you this slide, and I'm not going to talk through it in a huge amount of detail, but just to say that the data that I'm going to be telling you about came from this study, which I'll describe in a minute, which we referred to as Work Package 3, and samples were collected in that and then passed to both my lab and another lab in Belgium, and we've been doing the molecular phenotypes of the data. And then what we're hopefully doing today is Work Package 9, which is doing a bit of dissemination. So in Work Package 3, experiments were taken to generate and develop predictor phenotypes, and this was done in 6 research herds in 6 different countries. And within this study, feed composition and intake were monitored before and after calving. We had body condition scores, milk production, and quality information, and you'll be hearing a lot more about this from other people tomorrow. They had comprehensive metabolic profiling in milk, which actually I'm not going to be covering today, but different metabolites were measured in milk, and these were validated against more traditional blood biomarkers, including NEFAs, BHBs, but also importantly including IGF-1, which was the metabolic hormone I mentioned before. The mere spectra in the milk you will be hearing about. In terms of fertility, vaginal mucus scores were taken and endometrial swabs, and the fertility records were collected. Health records were collected, and all the cows were genotyped, and a subset we used for methane emissions in terms of looking at effects on the environment. So looking in a bit more detail at the experimental design, it focused on the first 7 weeks in milk. And during this period, twice a week samples were collected of milk, and these were analysed for the various things that I mentioned. But actually the bit that refers particularly to the work I'm talking about was the fact that blood samples were taken at 2 time points. So one was at 2 weeks after calving, which, as I've emphasised in the previous talk, we've known from previous studies is when you're in the sort of worst problems of negative energy balance, when issues are arising. And then a later one at about 5 weeks after calving. Phenotypes and clinical registrations were recorded. So the aim of the work that I'm just going to be describing to you now was to do RNA-seq analysis of whole blood. So that was taken from, so the blood that I'm going to be talking to you about was taken at this sample, whole blood sample, taken 2 weeks after calving. They were all Holstein Friesian cows, they were all in early lactation, and then we've related this to aspects of energy balance and fertility. So one of the things I think you need to think about, a lot of people now are taking whole blood samples. Clearly they're nice and easy. You can just stick a needle in the cow, you get the sample, comes out into a Tempest tube. But it is important to note that this is not a purified population. So within the white blood cells, we've got lots of different cell types, neutrophils, eosinophils, basophils. We've got some monocytes and some lymphocytes. And clearly, if you were a sort of proper card-carrying immunologist, you'd actually break these down into lots of different subclasses, which is something that we haven't done. So we've just got a snapshot of the whole lot together in the blood. These are some taken from a review paper, and they just give you an indication of how many of each of these cell types you would expect to find in a typical cow blood sample. So more neutrophils are the kind of overwhelmingly dominant one, but some of the others, and the relative proportions of those will also affect the health of the cow. So when we're looking at an overall expression profile, we need to bear in mind that we've got different cell types there, and we're not absolutely sure the proportions that they're present in the blood sample. But nevertheless, there's an awful lot of studies now, particularly from human medicine, that show that using this approach you can get useful information about the transcriptome of the individual that the sample was taken from. A little bit more on the methodology. So here we've got the Tempest tubes of what the blood is collected into, which contains its own preservative, so they're relatively straightforward to take. 229 cows from these 6 herds, and then they were processed for RNA-seq using an Illumina platform. And the cows we had, as I mentioned, we had body conditions scores, we had IGF-1 in the blood measured at 14 and 35 days in milk, information on milk yield and fertility, and I'll explain the fertility in a bit more detail later on as to how we actually quantified that. So this is just an overview of RNA-seq. I'm sure some of you probably know more about this than I do, but others may not, so I'll just go through it fairly quickly. So we start with our population of cells. We extract the RNA, it's then converted into cDNA, and then libraries are prepared and sequenced, so you end up with PCR amplification. And then you take a cow reference genome and you try to assemble all of these different bits of sequence onto the genome, and then it should be possible, once you've done all of that, to be able to work out various things from the transcriptome. I'm afraid this isn't terribly well in focus, but hopefully you can see what's going on. So you can measure the expression of all of the, well, hopefully throughout the genome, all the different genes within it. You can see in one go what the expression levels are. And that's what I'm going to be talking to you about today, is looking at the differential expression side of things. You can also use the information for other things. You can look at allele-specific expression. So that means that you can determine whether you've got differences in the produced by the part of the genome that came from the sire versus the dam. And you can also do a technique called eQTLs. But my colleagues in Belgium are doing both of those things, but I'm just concentrating today on this differential expression profiling. So a few more methodology details. So as I said, we used this whole white blood cell population to produce the libraries. These were sequenced at 75 nucleotides in length, single end reads, to reach 30 million reads per sample using Illumina sequencing. and then the files were mapped. And this is one thing it is worth mentioning, because actually very recently a new bovine map has come out. So we use this most recent Bos Taurus map to map these reads onto, and we have found that you do get some differences in interpretation depending on which genome annotation you actually work against. So that's something to, if any of you are interested in this sort of technique, you do need to sort of look at that. And then, as I said, we measured the differential expression between different groups. So one of the issues that we had to deal with in terms of trying to make any sense of this data was that we got samples from 6 different herds. These were from Afbys in Northern Ireland, Aarhus is Denmark, Italy, this one is in Belgium, Germany, and UCDs in the southern part of Ireland. And looking at the colour coding here, if you just use principal component analysis and just say, how do the cows behave according to country of origin? And you can see from the clustering of the colours that actually probably the main thing that's driving differential expression at a gross level is actually the herd of origin, which isn't what we're trying to look at, because we're trying to tease down as to within each of those herds, how is the energy balance and fertility actually affecting things. So it was necessary to remove the herd effect, and to do this we used this commercially available Partech software, and you can see that after removing the herd effect, all of the cows from the different herds are pretty much mixed up together. So we've managed to get rid of the herd effect in terms of analysing the data. It is actually interesting to note that there are these 2 odd samples that are out here, there, and these are the same cows that are now they've moved having done the change. But both of those cows were from the same herd, and they actually both had quite bad mastitis very early in lactation. So that is an indication that if there's a major kind of immunological event going on, then the white blood cells are not surprisingly behaving differently. One of the other things that we thought we should control for was the parity, because as I've said to you in the first talk, we know that parity does affect quite a lot of things to do with milk production and fertility. So we also investigated including parity in the analysis, and in fact, actually, if you just at a very gross level, just comparing this principal component analysis, it didn't actually make a huge amount of difference in this particular data set. But having said that, all of the data that I'm going to be telling you about going forward has actually been taken from the multiparous cows rather than the primiparous ones, because regardless of this, we thought that it was more biologically sensible to analyse them both separately. So that was just a quick whiz through the basic methodology behind the experiments. So again, quick pause in case anyone's got a question. No? If not, keep going. Okay, so the first experiment, at least the first analysis of the experimental data, was to look at the relationship between the IGF-1 and gene expression, because I've already hopefully made, going the wrong way, made the point to you in the first talk about how key circulating IGF-1 is in terms of the fertility of the cows after calving. So we have the entire genome sequence, so that's 20,000 plus genes, which we have the expression levels of. Some of them are expressed at very low levels, so we take those out. But of the ones that are expressed at sufficient levels to measure, you can do a correlation analysis. So every gene, the expression of every gene was correlated to the circulating IGF-1 concentration, which was taken at the same time as the blood sample was collected. And from that, we can see that we have some genes where there was a significant positive correlation. So the higher the IGF, the greater the gene expression, and others where it was going the other way, it's a negative correlation. So higher IGF, less, hang on. Sorry, no, that's, this is negative correlation, so I'm getting myself tied in knots now. Anyway, so some are negatively correlated and some are positively correlated. So using gene selection criteria of the significance within the correlation analysis of p less than 0.01, and we also had to have an R of greater than 0.3. Both of these are debatable, so you read different people doing this type of analysis, and people will take different levels of significance. And because we've actually got a large number of cows and a huge amount of data, you actually do get highly significant effects with actually very, very low correlations. But we wanted to take things that did have a reasonable correlation, so we took a cut-off of 0.3. And using those criteria, we ended up with 278 genes that fulfilled our definitions, and of those 42% were positively and 57% showed negative correlation. So the first sort of gross stab at doing the analysis was using Panther software, which just classifies the genes that were significantly different into, in this case, different molecular function categories. And here we've got them plotted in this format. So on the left-hand side, we've got the positively correlated genes, and on the left-hand side, the negatively correlated genes against a whole different numbers of molecular functions. And you can see, sort of just looking at these, that the main ones that are most significantly expressed are to do with binding and catalytic activity. And so bearing in mind that these are all white blood cells of some shape or form. So I was trying to sort of work out what to tell you today, and I've decided to just go in terms of picking up the stories of some of the genes that showed the highest correlation, so that they came up as being really positively or negatively strongly correlated with the IGF-1 concentration. And first of all, we have some genes that are in the IGF signalling pathways. So here we have a description of the IGF signalling pathway, which I'll just quickly run through. So we've got the green shows the IGF, which is circulating in the blood, and mostly it's attached to a variety of different binding proteins. And I've already told you that the expression of both the IGF and the binding proteins is going to be affected by the energy balance status. Once it gets to the cell membrane, then the IGF is removed from the binding protein, sticks onto the type 1 IGF receptor, and it can then activate a whole number of different cell signalling pathways. So coming down here this way, you've got RAS and ERK, and then those act as transcription factors in the nucleus, and that leads to cell proliferation. Coming down this way through the PI3 kinase pathway and ACT, you've got various possible actions of IGF on the cell. Having a high IGF will decrease the level of apoptosis, but increase protein synthesis and have positive effects on glucose metabolism. So within our particular experimental design, looking at the genes that were correlated with IGF, actually the one that was most highly significant was a new one on me, which I'd never even heard of before, which is this thing called IGF-2 BP3. But looking at doing literature search, quite a lot of people seem to have found this recently. So there's actually a lot of papers published on it in the last 2 years. And these have shown that it's, in various studies, in various cell lines and populations, it's positively associated with the IGF-1 receptor. And if you knock it out in these cell lines, then you get a decreased ability to respond to IGF-1. And people have shown that that's functional using migration and cell growth assays. So we know that that one is affected in this population of cows at this time after calving. And then we've got negatively associated with IGF-1. GRB10 is affected in this early part of the signaling pathway. PI3K3 is associated with the PI3 kinase here. And then the RABs and RASes are part of this signaling down to proliferation. So these are all, as I said, all negatively associated. So I think that does provide evidence that in the white blood cell population there are clear differences to not only in the earlier work that we did, we showed that we got differences in the circulating levels and in the binding protein, but this is able to take us a stage further and say that actually there's a lot going on within the cell that makes it less able to respond to the IGF that is there. Now, one of the other things that came up by looking at the most highly expressed genes were things to do with antioxidant activity. So in terms of nutrition and management of cows, these are also been known for many years to have a really important function in the cows in terms of immunity and health. And there are two key enzymatic mechanisms that protect the cows against oxidative stress. These are superoxide dismutase, or SOD, and peroxidase. There's a variety of peroxidases, but they include glutathione peroxidase. And the relevant thing here is that the glutathione peroxidase contains selenium, and the selenium is essential for its activity. So you can go back through the literature, and people have for quite a few years been supplementing cows with, very often there are supplements on the market that combine selenium with vitamin E. So it can be quite hard to separate out their relative effects, but they nevertheless are supposed to increase the ability of the cows to resist oxidative problems in early calving. So when we look at the genes that were differentially expressed, two of these were selenoproteins. So selenoprotein N is localized in the endoplasmic reticulum, and that's known to have a role in protection against oxidative stress, whereas selenoprotein P is the protein that transports most of the selenium in the plasma, and so that acts in a more extracellular way in terms of an antioxidant. And both of those were down-regulated, so the cows, as IGF-1 went up, these went down. And then there was also this gene, peroxyredoxin 2, which is another antioxidant enzyme. So again, there is evidence that in association with the changes in IGF-1 circulation, you're also getting changes in the ability of the cows to cope with oxidative stress in this critical period. So the third sort of little group of genes that were highly correlated with the IGF turned out to be potassium voltage-gated genes regulating potassium voltage-gated channels. And these were all positively correlated with IGF-1. So this particular picture of a T cell was taken from a really good, excellent review on immunity, where they were looking at ion channels in various immune cell types. And the summary from that is that divalent cations, such as calcium, magnesium, and zinc, have important roles as second messengers into regulating a lot of intracellular signaling pathways, whereas the monovalent cations, which include sodium and potassium, are mainly regulating membrane potential, but that in turn is indirectly controlling the influx of calcium in and out of the cells, and again having a really profound effect on immune signalling. So something else that was being altered in our immune cell population together with IGF-1. In terms of the group of genes that had the most differential expression were solute carriers of the SLC functions, and out of the 270 genes that we came up with, 10 of these were classified as solute carriers, so that was 3.6% of the whole. I'm not going to list all of these, but you can see from the colouring, the orange were the ones that were positive, the green were the ones that were negative. But just picking a few out, so this one, SLC12A2, is known to mediate sodium chloride transport, and that's very important in maintaining ionic balance and cell volume. This one here, CLC9A7, is mainly found in the Golgi, and it's critical for pH homeostasis in organelles. And then this one here, CLC11A1, is involved in transporting particularly iron and manganese in and out of the cells and in our metabolism, and these are known to be important in pathogens in terms of they are involved in the mechanisms of host resistance. So again, evidence that the activities of the immune cells are affected as the IGF goes up or down. And then finally, and just summary up a lot of genes quite concisely, clearly as we were taking an immune cell population, we were interested in actual which immune genes were correlated, and these have been summarised here. You can see far more in green than in red, so more of those were going down as the IGF was going up. And I won't go through all of these in detail, but they are between them playing roles in antimicrobial activity, for example, HP there, macrophage homeostasis, the complement system, responding to LPS, iron absorption, part of the interferon pathway, response to, we've got several interleukin receptors, TNF cytokines signalling down here, and the ability of the neutrophils to bind to the endothelium. So quite a range of different immune type activities have been altered in the cell population one way or the other. So I think that's my last one in terms of looking at the correlations. So that isn't directly looking at the fertility of the cows, but as I hope I convinced you in the first talk, the IGF-1 is probably one of the best indicators of the actual metabolic status of the cows, and we're showing that as the IGF-1 level changes, all of these gene expression levels change, and we know that the circulating IGF-1 is a really good indicator of fertility. And final point to emphasise on that is that we're sampling these 2 weeks after calving, but what's going on during that postpartum period is affecting what happens when you start trying to breed the cows, even though that's a long time later. Okay, I'll keep going. So for the final lot of analyses, this time we were actually focusing more directly on the actual fertility status of the cows in the study. So going through some of the methodology again, blood all taken 2 weeks after calving, and because we were only taking the multiparous cows, there were 172 of them, again from these 6 herds. We got the fertility and culling data, and it's, again, I showed you in the earlier talk that there are a bewildering number of ways of classifying fertility, and we decided to, for simplicity really, to go with a system that was, I think, originally developed in Australia, where they were calling them in-calf by. So rather than go with days open, it's easier to put them into these categories. So we have 4 in-calf by categories. So those that conceived within 100 days after calving, which we would say were the highly fertile ones. Group 2 took between 100 and 199 days. So those were not brilliant, but not disastrous. Group 3 took more than 200 days. And then group 4 were a bit of a mixed bag. Some of those never got served at all to do with a variety of different health problems. Others were served, but they failed to conceive. So the blood transcriptome is the same data as was analysed previously. And this time we did mixed model anivars using the Partex software. And we decided, because both of groups 3 and 4 contained quite a variety of reasons why these cows had such bad fertility, we decided to just focus on comparing categories 1 and 2. So these are the highly fertile and the not so good ones. And then the list of differentially expressed genes between those 2 groups were analysed using panther and cake pathways. So looking at what the actual fertility data consisted of, so here are our 4 different in-calf by groups. each with quite a large number of cows in them. So very often when you're looking at this type of analysis, you see maybe only sort of 3, 4, 5 animals per group, but we're talking about the smallest group still had 23 animals in it. So we've got large group sizes. The days open is what the definition for the in-calf buys were decided on. So they were actually, in this group, they calved between 42 and 99 days, in this group between 100 and 198. These ones, the longest one, and this is why I was telling you in the previous talk about the issues of waiting for cows to actually get in calf. So the longest time that it took from calving for one of the cows in this study to actually conceive again was 528 days. So that's generally after your project's finished, but nevertheless it's worth to kind of keep tabs on those cows. And then these ones were ones that we knew never conceived again. So in terms of body condition scores at 14 days in calf, after 14 days in milk, those weren't significantly different between the groups, although they were lowest overall in this group. By 35 days, although the differences look quite small, they were nevertheless significant. So the ones that took a bit longer had slightly lower body condition scores than the ones that conceived more rapidly. So they'd lost slightly more body condition over this initial period. And as I said, the days open is based on the definition of the categories. So it was obviously a lot longer in these ones than those ones. And they also require more services to conceive. So again, if an initial quick look at the transcriptomic data between categories 1 and 2 using Panther software, which is freely available. And so this time I've got 2 different of the analyses that we can look at. First one is biological processes, which is shown in this graph here, and the ones that had the most significant genes included in them were the grey ones, which is cellular processes, and the browny ones, which is metabolic processes. I was expecting, to be honest, more to be happening in terms of immune systems, because we're looking at immune cells after all, but I didn't mean to go back and change the colours on this. The pinky lot here is actually localization, and the immune you can really barely see. It's a very small sliver here. So that although genes get classified in different ways that go through into the system, but actually there wasn't an overwhelming kind of immune cell response that we might have anticipated, but the things that were coming up with these cellular and metabolic processes. And looking at molecular functions, then it's the same 2 that we found before, things to do with binding and catalytic activity. So looking at which pathways had the most genes in them, and here we've got various ways of classifying them. So we've got KEGG pathways, and these things called UP keywords and GO terms. And these were the ones that were most enriched in terms of our population of cells and had the highest significant values. And if you look at these, you can see that the term endoplasmic reticulum keeps coming up. It's in here. This one is short sequence motif, which is preventing secretion from the endoplasmic reticulum membrane again. So overwhelmingly, we start to get the message that actually what's going wrong in these cells is something to do with the endoplasmic reticulum. And then in the middle of all of this, and again, very significant, things that are going on in the mitochondrion, which is clearly the key part of the cell which is involved in energy generation. So just thinking a bit more about what goes on in the endoplasmic reticulum, which may not be where you initially think you're going to go when you start talking about fertility, but nevertheless, that's where we've ended up. So within the cell, the endoplasmic reticulum is responsible for taking the proteins that are generated and making sure that they're folded and modified as the cell wants appropriately. And the endoplasmic reticulum also has a key role in regulating intracellular calcium. And you have an issue that if the proteins don't get modified and folded properly, then they can't actually get out of the endoplasmic reticulum, so they build up within the lumen and they cause stress. So what should happen under normal circumstances is that you've got interchange between the endoplasmic reticulum and the Golgi, and some of these proteins are used for mechanisms within the cell, and some of them are going to be secreted. So when this goes wrong, you get this thing called the unfolded protein response, which is the modification that the cell makes to try to overcome the difficulties which it's experiencing. And it does various things to try to alleviate the situation. First of all, it inhibits mRNA translation, so it tries to stop itself from making more proteins, so you get a general overall reduction in protein synthesis. However, despite the overall reduction in protein synthesis, it will try to make more molecular chaperones which help with the folding, and it will also increase synthesis of proteins to try to increase the overall volume of the endoplasmic reticulum to give itself more space. And this involves various genes that are involved in fatty acid synthesis, which are important for the development of the membranes. So that's the first line of attack. But if all else fails and still you go on having problems, then the unfolded protein response activates apoptotic mechanisms which are associated with increased cytosolic calcium, and the cell itself will be killed off. So the message we're getting is that things are going wrong with the endoplasmic reticulum, which are clearly causing the cell stress. So if we look at the differentially expressed genes between the more and less fertile cows that are associated with the endoplasmic reticulum, these are nearly all coloured green, so they're nearly all lower in the cows with the better fertility. So these are the ones that conceived before 100 days versus the ones that took longer than 100 days. So we've got the other way of looking at it is that the less fertile cows, all of these things were being upregulated 2 weeks after calving. So there's a greater unfolded protein response, a number of these genes which are involved in regulation of calcium. These genes are known to be involved in vesicle trafficking between the endoplasmic reticulum and the Golgi. Protein synthesis, this is the only one that was actually regulated in the other direction. The lipid synthesis is probably to do with the membrane synthesis, sodium channel regulation, and apoptosis. So this wasn't probably where we expected to find, but that's one of the advantages of using this type of approach, that you don't actually, when you do PCR of target genes, you kind of go at the experiment with a preconceived idea of which genes you think may be making a difference, whereas here we've looked at all the genes within the genome, and the things that are really different between the more or less fertile cows are by and large genes that are regulating protein synthesis within the endoplasmic reticulum. So having got these set of results, this reminded me very much of some results that we'd had a number of years ago. So I'm now going back to this experiment that was done in Ireland, where cows were fed to produce animals that were either in mild or severe negative energy balance, and as I said, the animals were killed 2 weeks after the start of calving so that we could collect the different tissues. And one of the tissues that was taken from these cows was the spleen. So why is the spleen relevant? Well, it's a major player in the hematopoietic system. It's where mature lymphocytes are produced, and it's involved in the formation of antibodies. And it also acts as a reservoir for red blood cells and destroys worn-out red blood corpuscles, which was something else that we knew was different between the high and low negative energy balance cows. So this particular study, the spleen tissue was analysed using gene expression arrays, and lo and behold, this was done, this was published in 2009, so it was done about 10 years ago, but actually we found exactly the same types of responses in the spleen as I've just reported to you in the white blood cells. So the things that come up as being highly significant were things to do with oxidative stress responses, mitochondrial dysfunction, and here again we've got the endoplasmic reticulum stress pathway. So it's quite nice to have this kind of backup from the different experimental approach that's showing that when you go into negative energy balance, these are the things which are actually going wrong in the cows. So, starting to get towards the end and just trying to summarise some of this. So, I've shown you that in these early postpartum cows, we have metabolic changes which are required because the cows are short of glucose, so they shift over to using NEFAs as their major intracellular energy source. They have reduced signalling through IGF-1. This is partly because actually less of both of these hormones are produced, but also throughout the signalling pathways there are changes that affect the downstream signalling mechanisms as well. And that's actually particularly relevant for insulin. So there aren't, whereas we've seen really big changes in IGF-1 in the circulation, I haven't talked about insulin, but the insulin levels don't change in the blood to the same extent, but there are definitely effects on the signalling pathways within the cell. So when the cows are going through these processes, you get oxidative damage to important organs and tissues. I showed you pictures of the liver with all of that fat being accumulated. The spleen is affected, the white blood cells are affected, but also, and again, something that we have data on, but I haven't actually shown you today, there are changes in the oocyte and in the endometrium. And because the immune cell is compromised by all of this, then the cow is less able to respond to infections, so it's going to be more likely to go down with mastitis and more likely to get uterine infections such as endometritis. So there is no one reason why they're not fertile. It's very, very multifactorial, and all of these things going wrong have adverse effects on fertility at all levels. So the ovary doesn't function properly in terms of ovulating and producing good quality oocytes. The early embryo isn't properly nourished within the uterus and is less likely to survive. We also have changes in the oviduct and in the uterus. So all of those things make early embryonic development more likely to go wrong. So take-home messages that in order to overcome these problems, improving fertility really does require a very multifactorial approach. In the shorter term, the people who are good at managing fertility have an absolute eye to detail. There's no one aspect of the management that is matters. It all matters. You have to get it all right in every cow to get good fertility. So the nutrition has to be right. You need to be on top of health planning. The housing needs to be reasonable. And although I've put it up here, the actual nitty-gritty of inseminating them and so on is important, but that's actually downstream to all these other things that the cow itself has to be up to being in a good enough status that it has a sporting chance of conceiving if you do inseminate it at the right time. And as we'll hear more about tomorrow from some of the other talkers, our longer-term goal is to improve the breeding through careful genetic and genomic selection. However, I think we do need to be careful of using a black box approach in terms of selection policies because of the fact that it's led us astray before. In the 1950s, everyone thought that yield was the wonderful thing, and as we've seen, particularly in the Holstein breed, the overall selection in terms of improved yield had these negative effects on immunity and fertility. And as we get an understanding of how complicated things are in terms of these interactions between fertility and immunity and metabolism, we may inadvertently end up selecting for pathways that may be good for something and not so good for something else. So it is important to try to understand these pathways within reproductive tract and metabolic and immune systems, which are all interacting to regulate fertility. But as the advances in the last 10 years have really been absolutely amazing, so the rate of travel in terms of trying to understand the transcriptomics are really moving very fast, and I think we will get a better understanding of some of these going forward over the next few years. But my last slide is the absolute take-home message, is that a lot of the problems to do with fertility are before, during and after calving. So managing the cows during the peripartum period is absolutely essential if we're going to avoid problems later on. So the management of the dry cow is really what sets it up for the extent to the negative energy balance issues in that period 1 to 2 weeks after calving. And in all honesty, if the problems have arisen then, you can come along as a vet and you're asked to look at a cow 2-3 months later, and they say, what's the problem with the fertility? Can I sort it out? And really the answer at that point is no, because the problem's already arisen during this calving period. So this is when you really need to focus, and actually I'm quite glad in an institute that has a focus on nutrition, really getting the nutritional management and the health management right are what's going to help you have good fertility in the long term. So final slide is just the acknowledgements to GplusE. It's been a very interesting 5 years. We've had, as Michelle mentioned earlier, we've got all these different groups from different places. It's taken a while to get it all coming together, but I'm delighted to be able to share some of it with you today, and hopefully some of the papers from all of this work will start coming out over the next year or two and show some of the things we've found out. So thank you very much. I have a question, finally.
What are the insufficiencies for using white cells, white blood cells?
Yeah.
What are the insufficiencies? Could we get the same results using another approach to extracting and make those analyses?
Sorry, I'm not quite hearing.
What are the insufficiencies using the white blood cells?
So why are we using the white blood cells?
Yes.
Simply because, well, the main answer to that is that they're easy to collect.
Okay.
It's something that anybody can go along, stick a needle in, and you've got a sample of the white blood cell population. You can use it for other things apart from what I've been talking about here in terms of looking at you can sort of genotype animals and so on based on that. But in terms of the transcriptomics, you can't get samples very easily of the internal workings of the cow. And there's been, as usual, I think people working in human medicine are kind of 10, 15 years before us working in the animal science industry. And so there's now a lot of evidence from a whole range of different human diseases that looking at white blood cells, you can look at signatures in those that help you to distinguish between different human populations in terms of health.
Okay, I'm working in a system. It's easier to get the hairs, animal hair, and then when we make the same analysis using the hair to
hair.
Yes, animal hair.
Yes, I mean hair. There's no harm to use hair. It's just harder to get high yields of good quality RNA from hair. I mean, I've done quite a lot of hair. We've been doing a project on a rare breed where we were looking for a genetic abnormality, and that had a different set of advantages in that the farmers could pull out hair themselves, tail hairs, and then stick them in an envelope and post them to us. So in those circumstances, that was a good approach, and you can get DNA from those for genotyping.
But after extracting the DNA, it will be the same thing, like the white blood cells, to auto analyze to what you show us. It will be the same approach.
Oh goodness, I have no idea actually.
Okay.
I mean, the thing about the hair is that you obviously the only bit that has the cell in it is the actual follicle. So again, you're going to have a slight mixture of different cell types there. Presumably they are affected. I mean, I'm thinking now sheep and goats. I mean, sheep, we know that if they're in poor metabolic status, then it affects wool growth. So presumably there are going to be effects going on in the hair follicles. But that's a new one, so that's your experiment if you want to go and show that.
We'd be interested in examining the interaction between the genetic and management effect. For example, I don't know, using antibiotics. So effect on the phenotype outcome. If you use antibiotics, this is part of the management.
Yeah.
And this can change the genotype on the long run.
So the actual administering antibiotics isn't going to change. It's not going to change the genotype of the cow. It may be that you'll end up. I mean, I think one could argue that in production animals, because we've relied too heavily on antibiotics, then perhaps animals that are reliant on those to cure health problems have been allowed to persist in the breeding population. I mean, I think one of the things going forward, you know, one of the reasons behind GplusE and in all animal breeding populations at the moment is we're trying to get reduced reliance on antimicrobials. So we're all trying to produce cows that have fewer health problems, that don't need so many antibiotics. So that would be where we'd like to go. And I think what I'm showing you from this data is that if we don't manage the cows right, then we're really messing up their white blood cell population, so then they can't fight the disease themselves. Whereas if they were better managed, then hopefully they would be better able to mount their own immune responses. And there is quite good evidence for that, actually. I'm trying to think of actually one of my colleagues who's particularly interested in resistance to mastitis, and he's compared Holsteins, in his case with brown Swiss cows, and there are quite a lot of differences in the immune system in the brown Swiss cows that make them better able to resist mastitis. So you can keep the same cow in the same environment and the same management, and one breed will be more resistant than the other. So that clearly is where we all need to be going. Better understanding of what affects our susceptibility to these diseases, and then trying to breed animals that are going to be able to resist them.
I have one question.
Yeah.
You talk about eliminating the fixed effect for herd and parity.
Yes. Yeah.
Have you think to involve like the interaction between herd and season of calving, like a random effect in your model?
We didn't include season of calving in that. They were, I'm trying to think, they were all year round calving herds. I mean, to be honest, it's a perfectly valid question. And indeed, because of the way the samples were collected, it did go on over quite a period of time. So there would have been animals calving at different times of year. It's something to think about, but in these big Holstein herds with all year round calving, the season isn't as dominant an effect as it would be under some particular cows that are under a more seasonal calving pattern. Also, I have to say in Northern Europe we don't get sort of huge problems of heat stress in summer usually, which clearly in some parts of the world, you know, that's a bigger issue, but it's not some. This year was an exception. We had it very hot, but normally it wouldn't be an issue in the regions that the cows came from.
Thank you.
Right, I think
One question. It is well known that fertility is a trait with very low heritability.
yes,
For this reason, the genetic program is also very low.
yes,
In the past, or in near past, many countries used progeny testing to increase the accuracy of selections
yeah,
for this trait. And starting with 2009, in genomic era, some countries applied directly genomics.
yeah.
In United Kingdom, which is the situation? Do they still use progeny testing to improve fertility or directly use genomics?
Okay, I think that there was a slide where I looked at what was going on in the UK in the first talk. So we have several different indices and people can choose which one they want to look at. So some of the indices have sort of bits of everything, but then we have one index which is specifically targeted on fertility, and that includes some particular measures of fertility, like the days to first service and so on, and also it includes milk yield and body condition score. So that's, but I think that's only part of the answer. So that's actually based on daughter performance. But again, over the last, I mean, more recently than that, there is now a greater emphasis on genomic testing. So people are trying to pick the young bulls based on simply doing a genomic analysis without having waited to get the progeny testing results. And that, to be honest, you may hear more about that from Dara tomorrow. I don't know the proportion at the moment who are buying. I think the advice to the farmers is generally to pick some semen from bulls that have actually been progeny tested and some from the young bulls based on just on the genome analysis. So we're running both at the moment.
So, can I ask just a simple general question? So, we know we are facing a problem for Holstein cattle for looks like fertility trait. So just I would like to know your opinion. So if we do the crossbreeding for dairy cattle production,
Yeah.
so do you think it will be the long-lasting for dairy production system for improving the fertility trait for dairy cow?
Yes, I mean certainly quite a lot of the UK farmers are, and elsewhere in the world are using crossbreeding. There are some crossbred with Jerseys. There's a lot of imported Scandinavian genetics at the moment as well. I think that the problem is getting a kind of balanced cow. So the first cross is fairly uniform, and then you sort of have to decide what to do after that. With more extreme crosses, it affects the size of the animals and then the design of the buildings and so on. So it can get quite complicated. But nevertheless, the main farming type in the UK who are going... So the UK system has been predominantly all year round calving, and the bigger farms in particular going with Holstein genetics. But more and more farmers now are going for seasonal calving where they can make better use of the grass. So you have a lot of spring calving herds, and a lot of those are using crossbred. So they're going for a smaller animal that can cope from grass and is more fertile and hopefully lasts longer. And those people are using crossbreeding. And I think that will as far as I know. I mean, that's actually been a growing trend, so I think that will go on, yeah.
Okay, thank you.